Continuing on now with the third, I think we're on the third part, yes, of chapter 6 here. So we're going to now trace the signal from the eyes back further into the brain after we have now finished talking about what's happening at the retina. And we had finished last time by talking about one of the ganglion cell pathways taking us to the so-called suprachiasmatic nucleus. And I suggested that it is named because of the place where it sits just above the optic chiasm. But what in the world is the optic chiasm? And I think, well, I have some text here. A better thing for me to do would be to go to the next picture here. Remember, the Greek letter chi is an X, right? So that's really what we have here, is that if we look at this diagram, we can trace the optic nerve leaving the back side of each eye, and we see that they come together and partially cross over to form an X shape. And so that crossing over is the optic chiasm. What is not shown here would be the hypothalamus sort of sitting right above that, so that there would be additional nerve paths connecting for the ganglion cell pathways to the hypothalamus and the SCN or suprachiasmatic nucleus within it. But for the rest of this, we're really going to not be too concerned with that particular pathway. We may be more interested in the pathway that takes us ultimately back to the occipital lobe of the brain, where conscious visual perception occurs. But the first thing we want to understand is how the brain is mapping the visual field. You are familiar with the concept of cross-laterality in the brain, right? And we normally think of things like, okay, my left hand and sensory information from my left hand is processed on the right side of my brain, and conversely, right hand to left side of the brain, right? But with the eyes, it doesn't quite work the same way, because you might think, oh, does that mean the left eye goes to the right side of the brain and information from the right eye goes to the left side of the brain? And the answer is no. And it's not about how the eye maps to the brain. It's about the external visual field. And so what we see in this particular picture is how the left visual field is color-coded in red. So that means everything to the left of the person's nose is the left half of their visual field, and everything to the right of their nose is now the right half of their visual field, which is color-coded in blue, right? And we see that mapping onto the retina. One thing that we might need to remember about the retina, and I know we didn't talk about really how optics work in the eye, like the projective properties of the lens, but you might have learned this in another class prior, that the lens inverts an image when it projects the world onto the interior lining of the eye, the retina. It is flipped upside down and also flipped left to right. So in this case, what you see, like look at the right person's right eye. The right side of the visual field is mapped to what would essentially be the left side of their eye, which is also referred to as the nasal side of the right eye because it's closer to the nose. That's what I also have on the previous slide. And so again, we also see on the left eye, the left visual field is mapping to the nasal side of their eye, color-coded in red. So the nasal side of each eye is processing opposite sides of the field of view. For the right eye, that's the right field of view. And for the left eye, it's the left field of view. And then it's the temporal side, that is the outer half of the retina, that processes the opposite side. So for the right eye, the temporal half of the retina is receiving information from the left side of the body, the left visual field, left of the nose, right? And again, for the left eye, it's receiving the right half of the visual field. It goes to the temporal side. And on the previous slide, that's really what I have mapped out here in words, but also about what happens to the axons. So let's go back to the picture here. What we see is that the left half of the visual field from the right eye, as it leaves the temporal side, it stays on the right side of the brain. So as it maps back to the optic chiasm, it continues further back, going to there labeled the lateral geniculate nucleus, whereas the nasal side, which is the right visual field, crosses the chiasm and goes to the opposite left side of the brain. And then for the left eye, something similar is happening. The nasal side crosses the chiasm and goes to the right side of the brain, but the temporal side stays on the same side. So that's what we're seeing here, is that it's the nasal side that swaps sides, and then it's the temporal side that stays on the same side of the brain. And what this does is this results in the blue, in this case, the right visual field ending up on the left side of the brain, and the red is the left visual field ending up on the right side of the brain. But where does it go back after the optic chiasm, which is really just a crossing point, and there's nothing else particular of interest happening there? It goes to what is labeled there as the lateral geniculate nucleus, or LGN, and that is a part of the thalamus. So this entire organ here, we see this showing the little red bulb here and the blue bulb over here, as two LGNs, right? There's a left and a right LGN. This entire organ that they are a part of in the middle here is the thalamus, and the thalamus is this relay center that receives various inputs from various sensory nerves and then routes them to the appropriate part of the cortex. And since we're talking about vision here, and of course it contains many nuclei, and that's again a cluster of neurons that works to do a particular job together. So in this case, there's a visual nucleus, right? And there are auditory nuclei and tactile nuclei and so forth in the thalamus, but here we're talking about vision. So we see that the left and right lateral geniculate nuclei send their optic radiations, which are just nerve tracts. Now, we're not going to call it a nerve anymore, right? Because a nerve is a part of the peripheral nervous system, but a bundle of axons in the central nervous system now is a tract. And so these optic radiations are tracts, taking us back to the occipital lobe, where we're going to have different levels of processing. It starts in what is called V1, which is just shorthand for primary visual cortex. But let's find out what also is happening on these pathways as we travel back to the away from the eye. Well, one of the more important pathways that we're going to be spending most of our time talking about is called the geniculostriate path. And here this refers to the geniculo is referring to the lateral geniculate nucleus, so that part of the thalamus, and the striate cortex. Striate means striped, and this is a name that is used to describe the visual cortex because of the fact that when you look at the visual cortex sectioned under a microscope, it shows heavy bands or stripes because of the changes in the density of neurons in different layers, and so it has a very heavily striped appearance. This is the pathway, the geniculostriate path, that is used for conscious visual perception. What we're going to see later, once we look further into the occipital lobe and what's happening there, is that the information in conscious visual perception is split into these two separate streams that are sometimes referred to as the Watt channel of visual processing and the Ware channel of visual processing, but more on that later. We also have the tectopulvinar pathway. And so in this case, this is partly going to the, remember we have the midbrain that contains the tectum and tegmentum, right? And so the tectum here contains the superior colliculus, and that is the part that receives visual input that I had suggested before, may regulate blinks and eye movements and things like that. So in fact, this pathway is part of that. It helps regulate eye movements and visual attention, making sure that our eyes are pointing at things we're trying to attend to, for example. It also travels it communicates with the thalamus, but it's a separate location in the thalamus called the pulvinar. And it does interact with other higher level conscious visual processes in the parietal lobe and the temporal lobe. So there's quite a bit of complexity here. We don't need to worry about it too much. Just understand that that's a separate channel of visual processing that kind of deals with more like the conscious and also sometimes involuntary control of eye movements directed by attention. If you've studied and learned about eye movements before, you may have learned about bottom-up and top-down processes in eye movements. And I think this idea that there can be some involuntary kind of stimulus-driven attentional processes here is the part that involves some of the lower level parts, and the parts that involves conscious controls when we might talk about top-down control of eye movements. And then third here is the retinohypothalamic path, and this is just another name for the pathway we were talking about before of these light-sensitive ganglion cells that communicates with the suprachiasmatic nucleus, and that pathway is called the retinohypothalamic pathway. But the geniculostriate path takes us through the LGN of the thalamus, and that's where we want to go next. As I already noted, there is a right half of the thalamus and therefore a right LGN and then a left LGN. The LGN itself is organized into 6 layers. And as we see here, layers 1 and 2 contain the magnocellular pathway. That's from the rods. Layers 3, 4, 5, and 6 are a part of the parvocellular pathway. We can kind of see that in this drawing. So again, we see the mapping of the retinal projection to the through the optic chiasm. We have the nasal side crossing over and the temporal side staying on the same side of the brain. And then it goes to the right LGN. And here we see layers 1, 2, 3, 4, 5, and 6 preserving these magno and parvocellular signals coming from different parts of the retina. But also the left eye, right eye distinction is preserved in different layers of the LGN as well. So you can see that there, that the left eye is going into layers 1 and 4 and 6. That's coming from the contralateral eye, the contralateral side of the vision there. And layers 2, 3, and 5 are coming from the same side eye. That's called ipsilateral, right? So lots of layers of organization there. Some are based on which eye the signal is coming from. Some are based on which visual pathway, magnocellular or parvocellular, right? This is helping to maintain some organization and separation. One of the reasons why we want to maintain the left versus right eye signals and not sort of lump them all together is because one of the things that we do with our 2 eyes is we use them for depth perception, right? And what that means is that in order to have what we call binocular depth perception, we have to realize that each of the 2 eyes has a slightly different perspective on the world, so that the images the 2 eye received are actually shifted a little bit because the left eye is shifted a couple of inches away from the right eye. And the difference in the retinal image of each eye is called retinal disparity, or sometimes called binocular disparity. And the spatial differences in those 2 images can be compared with each other to establish the distance to an object. And so we need to maintain a difference in the left eye, right eye signal as we map everything back to the brain, so that that disparity can be evaluated by the brain and used for depth perception. Now, let's talk about these visual streams that I had said that when we talk about conscious visual perception, there is a where pathway or where channel and a what channel. These are also referred to by their anatomical locations as dorsal and ventral streams. These terms like streams or channels, these are really just terms that are meant to describe the flow of information through different locations of the brain. So the information is flowing through this pathway or flowing through a channel of nerve paths. And so what's happening is that in the geniculostriate path, as it maps from the LGN back to the visual cortex and the occipital lobe, there are some of the parts of the occipital lobe that seem to be dedicated to processing spatial information, right? Where is the object? How far away? And one of the things that we would want to do when we're talking about trying to figure out where an object is obviously we want to use that information so that we can interact with it correctly. If I need to walk towards it or reach for it and physically interact with it in some way, this would entail that I need to integrate visual information about the spatial environment with body information, right? Where is my body in this same spatial environment that I can see with my eyes? And I want to integrate vision with body information. The body information comes to the parietal lobe, right? So this is now referred to as the dorsal visual stream because of the idea that this channel of information is moving from the back of the brain, the occipital lobe, up to the top of the back of the brain where the parietal lobe is, right? Separately from that, it's called the ventral stream because now a separate pathway of information takes us from the occipital lobe down into the inferior temporal cortex, which means down to the bottom sides of the brain. This appears to be a separate pathway that is involved with conscious visual perception of what an object is. That's why we call it the what channel. What the point of this here is that there are different neural pathways and processes involved in, I have my coffee cup in the picture here as an example, right? So my ability to look at this object and say, that's a cup, involves the ventral stream. And that is an entirely anatomically distinct process than my ability to reach out and pick it up and bring it up and sip from it. So recognition, and that means involves what is it, what's it called, that is separate from the interaction of where or how do I reach for it and pick it up, right? So, but before we get too much further into that kind of deeper processing, let's look at what's happening in the occipital lobe itself and how the visual cortex functions. So again, we're going to talk about that genicular striate pathway from the LGN to the visual cortex. V1 is the first area. That's why it's called V1 in primary visual cortex, because it's the first part of the occipital lobe to receive the input from the LGN. But there are many other areas, V2, V3, V4, and so forth, right? As I've already noted, we have these layers in the cortex, 6 layers, right? And we've learned about some of that in an early chapter as well, right? The architectonic maps of the cortex, right? And these layers, as I said, create that really striated or striped appearance. The different layers, by the way, of course, preserve the magnocellular and parvocellular pathways coming from the LGN, and therefore are encoding different aspects of information about brightness, color, and shape, and things of that nature. So, of course, it might make sense as we, think about this a little bit, that information from the cones and information from the rods might be differentially useful when it comes to that where versus what channel, right? And so the M and P pathways here are partly relevant to distinguishing what is the where channel and what is the what channel. Something that's also interesting here is that these columns and so we can talk about layers, the 6 layers of the cortex, but now there are also vertical columns here, and these are called cortical columns. And each one of these columns is corresponding to a specific location somewhere on the retina. So it's called a retinotopic map, is we're taking the shape and geographical mapping. This is where the topic word comes from. Topos means space or shape or something like that, right? And so the shape of the retina, the organization and layout and mapping of the retina is preserved with the organization of these, first of the actual individual layers within the LGN, but then as that maps to these different columns of cells in the V1 area, then the retina gets mapped out in V1 as well. And so, like I said before, when I think about how the cones align kind of one-to-one to some of those bipolar cells and ganglion cells, then that spatial difference in a distribution of light across the fovea, for example, is also mapped out in adjacent columns in V1. Here we can kind of see some of that. So on the left side, again, we see the different layers of the LGN mapping M layers and P layers, right? And also left eye, right eye stuff. And then we see that actually preserved in these first in the columns, right? So we see left eye column, right eye column. And we're also seeing different layers of the visual cortex. And of course, different layers from the LGN go to different layers of the V1. The first layers that receive input are actually layers 4, right? So those are labeled there in Roman numerals as 4C alpha and 4C beta. And we don't necessarily need to worry too much about the differences there. But what will then later happen is that there will be other communication with some of those higher and lower layers. The section in the middle there just kind of helps us get maybe a visualization of why it's called the striate cortex, because of the way in which these columns are the density changes enough that you get this very obvious striate appearance under a microscope. But why are they called ocular dominance columns? So remember what I said earlier about this idea that we want to preserve left eye, right eye stuff. So one column might be processing information from the left eye, and another column is processing information from the right eye. And that's what we mean to say it's an ocular dominant, right? So one column is dominant from the left eye, and the other one's dominant to the information from the right eye. And the differential signals within adjacent layers can be combined and compared to evaluate the disparity, right? And that disparity is the difference in the left eye view versus the right eye view that can give us that good binocular depth perception that we want to have. Here, a few more pictures here that are showing us some of the complexity. And there's so much complexity in the visual system that there's more than we can really try to even think about covering here. But I want to give you just a little bit of a view. As I said, V1 is primary visual cortex, and it's the first area to receive input from the LGN, specifically in layer 4 of V1. But think of the visual cortex here as really a bunch of concentric circles. And so in the middle, and you can see this slightly better on the right-hand picture here, B labeled lateral view. You see V1 color-coded in pink there as kind of like this central little oval shape on the very, very back of the brain. And then wrapping around that like a donut in orange is V2. And then wrapping around that in the darker green is V3 and lighter green V3A. And then in blue, V4 and red, V5, smaller, not fully wrapping around. But the idea is that these layers are kind of spreading outwards, right? So it sort of starts in the middle and then it's in V1, and then it just spreads out. And as the information spreads outward from the occipital lobe, some of it, as we had noted, is going to travel up the back of the brain into the parietal region. And that's going to be our dorsal stream. And then some of it's going to travel down towards the bottom of the temporal lobe. And the bottom of the temporal lobe is called the inferior temporal cortex. And that's going to be our ventral stream, or the Watt channel. Now, this is more of a kind of a flow chart of visual processing. And again, there's a lot of information here that I'm really not too concerned that you're going to need to try to understand all of the details of it. But I want to just to give an idea, a taste of what's the complexity that's happening here and how much we might know, but maybe also what we don't know about it, right? So what we see here is, again, on the far left, we have the LGN sending information to V1. What is V1 doing? Well, we talk about blobs and color, right? So what is that? Well, what we're trying to do here is we're trying to organize the visual field into meaningful little groups of patterns. And so we might get little sections of a shape of an object here, a line here and a line there. And so we'll call those blobs for now, right? Because they're not fully defined shapes and objects just yet, right? So we get some information about form and movement as well, possibly happening in V1. And then we're going to send now some projections, some axons to V2. And V2 can evaluate whether or not we're dealing with forms that are thick or thin, right? And so all of these different combined pathways are going to be evaluated in V2. This is called extrastriate cortex because now V1 is just the striped part. Now we're outside of that. So that's what extrastriate means. And then from there, it doesn't just go 3, 4, 5, 6 in order, but rather it goes to, some of it might go into V3 for form or dynamic form. Some goes to V4 for extra color and for processing. V5 is for motion processing. And then from there, the dorsal stream, it will take over that form and motion information take it to the parietal lobe so that we can process the where and how information, and the form and color information is going to happen through the ventral stream so that we can take that to the temporal lobe for recognizing what it is, right? So the complexity for how this information is kind of split into these different meaningful pathways and taken to other parts of the brain is quite a bit. And on the bottom there, you can kind of see those arrows telling us where those different pathways go. It shows that red arrow going up the backside of the brain. That's why it's called the dorsal stream. And the blue arrow is taking us down into the temporal lobe. Now, I want to say a little bit more about what I had noted before about the retinotopic map and how we provided information to the brain about where an object is. So we've already understood that the visual world is divided into a left half and a right half, and that's mapped onto different halves of the retina and how that maps to each side of the brain through the optic chiasm and on through the LGN. But going back to that topic for a second, one of the things that we want to understand is what these ganglion cells are doing on the retina. And we had mentioned this before, this concept of them having a receptive field. And so a receptive field refers to the idea that a single ganglion cell is receiving input from several other bipolar cells, which in turn is monitoring even more rods and cones. And so all of the activity of a whole bunch of rods and cones essentially funnels down through the bipolar cells, and then all down to maybe one single ganglion cell, which sends a signal back to the brain, right? And so what we would say now is that that particular ganglion cell essentially is monitoring some region of the retina. And we're going to call that its receptive field, right? What region of the retina with all of these photoreceptors is that particular cell watching and receiving input from, right? Something we also want to understand, though, is that these receptive fields of a given ganglion cell are not exclusive to that one ganglion cell, right? So it's not as if the retina is really just subdivided into a bunch of little receptive fields that are mutually exclusive and only go to one single ganglion cell, but rather the ganglion cell receptive fields are overlapping each other so that any one rod or cone somewhere on the retina might be part of the receptive field of 3 or 4 different ganglion cells, right? And so they have these overlapping receptive fields. And then what happens is that if we think about how the ganglion cells send information back to the LGN is essentially monitoring a larger group of ganglion cells, right? So many ganglion cells converge down to maybe a single LGN cell. And again, though, there's still some overlap of these receptive fields. But what we're looking at here is something of a funneling of information from millions of rods and cones down to some smaller number of bipolar cells and ganglion cells to some smaller number of LGN cells, right? And so we're kind of funneling down all of this information. And ultimately, though, we map it back to V1, where we have these ocular dominance columns that are mapping out adjacent areas based on the organization of all of those receptive fields that began with those ganglion cells. We also don't want to forget that even though we have subdivided the left half and the right half of the visual field onto the 2 sides of the brain, we don't see the 2 sides of the visual world as independent visual experiences because the corpus callosum helps the left and right side of the brain communicate with each other. Although if we wanted to get into some of the research on people who have had a corpus colectomy, you sever the corpus callosum, which is something that may need to be done in very severe cases of epilepsy. What you'll oftentimes find is that anything that's presented on the left side of a person's field of view, which travels into the right side of their brain, they may not be able to interact with it very well with their right hand because the right side of the brain is actually controlling the left hand, right? So they might be able to reach out and pick it up with their left hand because the right side of the brain has this information. But in order to operate their right hand, that information would not be able to make it to the left side of the brain because their corpus callosum has been severed. But in a normal intact brain, the 2 halves of the brain interact and share this information. This diagram helps us maybe try to visualize that kind of funneling down, right? So we have all of these many retinal ganglion cells that have these small receptive fields all across the retina. A grouping of those retinal ganglion cell all now mapped to a single LGN, which now has a large receptive field when we map it back to the retina, right? And then some grouping, some functional grouping of LGN cells now maps to a single cell somewhere in V1. Now, when we think about the cells in V1, and actually not just in V1, I guess we're going to first talk about the ganglion cells. I forgot where I was going with that. So we want to understand the properties of these receptive fields, and therefore understanding the property of the receptive field helps us understand the nature of the information that the ganglion cell sends back to the brain. So what we see about the retinal ganglion cells here is that the shape of their receptive fields is a bit like a donut, right? It's got this inner circle and then an outer circle that surrounds it like a concentric circle around it, right? And what we find is that the very typical case here, there's some variation and some exceptions to every rule, of course, but very commonly we see what's called an on-center, off-surround receptive field. And so this is what we see on the left, is an on-center, off-surround ganglion cell. So what does that mean? What that means is that if we take a light stimulus and we shine it somewhere onto the eye, onto the retina, and it lands within the center part of that ganglion cell's receptive field, that's going to provide excitatory input to that ganglion cell. And that's what we're seeing in this little graph over here on the right that shows those little purple lines here. These are graphs that are meant to depict neural firing rates. And so what we're looking at here is that at time 0 up to time 1, there are 4 little spikes, and those 4 little spikes correspond to action potentials that we might record from that ganglion cell. But if you remember from earlier lectures, neurons have what's called a background neural firing rate, right? They may just occasionally leak a few potassium and let some sodium ions, right? And so there's this kind of spontaneous firing rate, and that's what we're seeing here from time 0 to time 1 is 4 spontaneous action potentials out of that ganglion cell. But what happens as soon as we shine a light onto the on center of that receptive field, we get this really rapid increase in the firing rate, so that now from time 1 to time 2, and we have to count the lines there, but 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, like 15, I think, of action potentials occur. So we've ramped up the firing rate from 4 per second to 15 per second now by shining a light on the center. And when we turn the light off, it goes back down to its spontaneous 4 times per second firing. But notice here that if we shine a light instead on the bottom left here in the outer ring, which is called the off area of the receptive field, we see something else happen. We go from the 4 spontaneous action potentials to now in our 1 second from second 1 to 2, just a couple of action potentials. So now we're looking at an inhibitory response, right? This ganglion cell is being inhibited by the presence of light in that outer ring. And then once we turn the light off, we'll go back to our spontaneous firing rate. Remember when we talked about that crosstalk from the amacrine cells and the bipolar cells? This is exactly what's happening here, right? What's happening is that wherever we shine a light, we're going to excite the rods and the cones. But what's happening is that the ganglion cell is kind of in the middle of all of these concentric circles, and it's going to receive excitation from whatever is right there in the same region that the ganglion cell is found. But there's this lateral inhibition, so that if there's any excitation in any neighboring regions in this little circle around it, that's going to travel laterally to inhibit this particular ganglion cell, right? So that's what is happening here. And so now this provides information that's useful to the brain, because if this neuron is excited, it knows that there's a light stimulus in one specific region. But if the neuron is inhibited, it knows that that light is somewhere else just off to the side, right? Some receptive fields are the converse. So on the right-hand side of this drawing, we see what's called an off-center on-surround, and you can infer that it's basically the same thing, but now flipped, so that now any light falling in the middle creates an inhibitory response, and any light falling in the outer ring creates an excitatory response. So in this case, instead of lateral inhibition, we actually have lateral excitation, which also is possible, but further complicates the total number of different kinds of signals that we're talking about here. And now, as I said, these receptive fields actually overlap each other. So now we are looking at this kind of a very complex mapping of many different receptive fields from many different retinal ganglion cells, all monitoring overlapping regions of the retina. Some have on-center off-surround organization, some have off-center on-surround organization, but the combined excitation and inhibition of all of these ganglion cells is now mapping back to the LGN, and then of course mapping back to V1. And of course, we're going to compare to these receptive fields to say, okay, is there some sort of a signal here that we can interpret as a presence of light in one place versus the absence of light in another place? And then we can figure out where's the light area, where's the dark area, and this can help us map out edges, right? And that's, of course, what we need to do. We need to map out edges, because an edge can turn into a shape, and a shape can turn into an object, and we can figure out what it is we are in fact seeing. But how do we map out shapes? And this is where we want to look at V1. Now we're going to take this signal back to V1, and the neurons in V1 have a receptive field as well. But the shape of this receptive field is not this donut-shaped receptive field. It's more of a rectangular receptive field. And what we see is that the cells in V1, especially in layer 4A of V1, have receptive fields that are specific to these rectangular bars. And some of them have an orientation that we might call a horizontal preference. So what we're seeing here is in the left-hand side of this drawing, we see that when there's no light, we have an on-center bar, and above and below it would be two off bars, right? If there's no light, we're just going to have our baseline spontaneous neural firing rate. But if we shine a light that is a horizontal bar across the retina, and we present that horizontal stimulus to the retina, then this neuron increases its firing rate quite rapidly. But if we rotate that light so that instead of being horizontal, it's some other orientation, like diagonal in this case, or vertical even, then we're going to get some activation in those inhibitory off regions. And so we're going to get a decrease in firing rate, no response. Some of these cells in V1 might have more of an oblique or diagonal preference. So in this case, you see the baseline response. When you present a horizontal stimulus, you get inhibition. But when you present the diagonal stimulus, you get excitation, right? So what we would say here is that these so-called simple cells are essentially neurons that like to respond to a stimulus depending on its orientation. So when you're looking at a horizontal line versus a vertical line versus an oblique line, there are going to be different simple cells in V1 that preferentially respond to these different neurons, or to these different stimuli, right? And so in this way, we can start mapping out the presence of lines in the visual field that were detected by the retinal ganglion cells and LGN cells, but now we know the orientation of these lines based on what the simple cells are telling us. And now the complex cells can also provide information about the movement of this line, right? So if the line is moving left to right, or right to left, or bottom to top, to up and down, right? The complex cells can provide information about the movement. And then we also have what are called hypercomplex cells, or sometimes end-stop cells, because what these tell us is that if that line is a specific orientation and moving in a specific direction, but if it stops in a specific location in the visual field, that might excite this neuron. But if that line continues on into another region that's called the inhibitory region, then it's going to start sending inhibition to this cell. So we could say that these so-called end-stop cells, we're calling them end-stop because it tells us where does the line stop. And usually that means that there could be some sort of an intersection between two lines in the visual field, such as think of the difference between like a letter T, right, where one line intersects another and stops right there at that point. That might activate a specific hypercomplex cell, but if it continues on through, then it's no longer a T and it becomes maybe a plus sign or something, right? And then that's a different kind of an object, and therefore it would inhibit this particular cell, but maybe excite a different cell. Here we see the organization of the layers in visual cortex, so that we can see that some of the different layers and some of the different columns contain different simple cells that prefer these different orientations. So as we go from these columns left to right here, these adjacent columns are mapping out different lines on the retina of different orientations. So we got, vertical line detectors, and then a slight oblique preference, and then even more and more and more till we get to the middle where we have more of a horizontal orientation, and then it kind of rotates back to the vertical at the end. And then we see the ocular dominance columns on the right, where of course the pink columns are for one eye and the blue columns are for the other eye. So of course we're going to map out those slight differences in the left eye, right eye view, so that we can still evaluate the differences there for depth perception. Then we want to finally get higher up into the brain so that we can get information about what it is we're actually seeing. So let's talk about the ventral Watt channel. So now these are projections from the visual cortex, the occipital lobe, into the temporal lobe, specifically, again, the inferior temporal cortex, the lower parts of the temporal lobe. And what we see there is that there are columns of cells in the temporal lobe that respond to different kinds of objects in terms of categories. So if here we're seeing, for example, categories of maybe simple geometric shapes, categories for body parts, hands and faces, categories for objects like fruits and things of that nature, right? So there's some meaningful grouping there. In particular, right, that face column is very interesting because what we know is that there's sometimes people who have damage to the temporal lobe of the brain, maybe because of a stroke, that specifically affects the face perception columns in one region of the temporal lobe. And this impairs their ability to recognize human faces. It's called prosopagnosia, right? In fact, in general, any brain damage that interferes with conscious perception of what an object is called an agnosia, right? So whether it's prosopagnosia, where you can't recognize faces, or sometimes a much more profound agnosia that just prevents you from being able to name anything, right? You look at the cup and you just don't know what it is, right? I can't tell you what it is, right? It's also worth noting, and I know we haven't really gotten this far into the class, but also found in the temporal lobe is auditory cortex. And what's important about that is that that's connected to something called Wernicke's area, which is part of language comprehension. And that, of course, includes our storage of the meanings of words. So it might make some sense that the visual perception of what an object is happens in the temporal lobe, which is where we also find our memory for the meanings of words, because of course, being able to look at something and say that's a cup is pretty important when it comes to not just recognizing the shape of the object, but connecting that to a word somewhere in the brain. What's a grandmother cell here? Well, we might imagine that if we could think about the fact that some of these cells in the visual cortex are cells that seem to prefer to respond to specific lines of a specific orientation or lines that stop somewhere in the field of view, that what we could start doing is we could start tracing this complexity further up some hierarchy so that we might find, for example, shape detectors. So like we see this column here that has a square and a triangle on it. Does that mean that there's a neuron somewhere in this column of cells in the temporal lobe that will fire every time I'm looking at a square, and that somewhere near there is another column that will respond every time I'm looking at a triangle, right? And then we might say that I have square detectors and triangle detectors, and that if I want to talk about reading, for example, does that mean somewhere in the brain I might find a letter R detector and a letter S detector and a letter W detector? That would mean that there has to be a very specific neuron somewhere in the brain whose sole job is to be activated every time I'm looking at a specific object. So that means that not only are there R, S, W, T, U, V detectors, and there are also dog detectors and cat detectors and bird detectors and cup detectors and table detectors, but there would be face detectors, and there would be a specific neuron for recognizing your grandmother's face, and the same neuron would be activated every time you see your grandmother. But what we find is that even though we know that there are these columns of neurons that seem to be dedicated around processing certain categories of objects like faces and forms, no one has ever really found this so-called grandmother cell, right? A single neuron somewhere that was always active when you're seeing a specific person's face or when you're seeing a specific kind of an object. So how actual complex object recognition occurs is not yet fully understood at this neuroscientific level, because we cannot really say that there's a specific cell somewhere whose job is to recognize that thing, right? We do know that neurons here are influenced by learning, right? So of course, as we learn new categories and we learn to recognize things in different ways, that we see the neurons here functioning very differently as a function of our, say, perceptual expertise in recognizing different objects. But what's really happening there at the level of recognition is not yet at all very clear. Let me skip past this real quick because I want to go towards the section on motion and action, and I'll come back to the color part for now. Well, the thing to say about color is that color is being processed on the Watt channel primarily, because color is perhaps very important for recognizing objects, because we might know bananas are yellow and apples are red, right? We've already covered the so-called trichromaticity theory, so I'll just go through this quickly. So that's the idea that they have those three different cones, and that the proportional ratios of activity between the three cone systems provides information about color. We talked about color blindness briefly. There are some color after effects that this cannot explain, like why, for example, if you stare at something that's green for a long period of time, you'll see an after image that is red. There's other processing that happens in the LGN, though, that explains these color dominance channels. But I'm just mentioning color here because it's part of that Watt channel here as well. Now, how does the process in the LGN work for color? Well, in the LGN, we have what are called opponent process channels. And that means that a channel of cells within the LGN can either process red or green, but not both at the same time. And so what's going to happen is that if you're looking at something that's green, that's going to excite this channel it's going to excite the green half of this channel, but it's also going to inhibit the red half of this channel. And then when the green stimulus is removed, the green half of the channel is fatigued and it's trying to recover. All of those neurons are going through a refractory period. And meanwhile, because the red half of the channel was being inhibited, all those neurons are rested and they're refreshed. And so they're going to return to their baseline firing rate. And so after the green stimulus is removed, what you're going to see is a red after image, because the red neurons, or the so-called red neurons, the ones that are processing information for red, are firing faster than the green neurons, which are currently refractory. And so you see that red after image, because actually how this channel processes information is about determining which half is more active at any given moment. And of course, when you're looking at something green, the green half of the channel is more active. But when you take away that stimulus and the green half is fatigued and the red half is now recovered back to its spontaneous firing rate, now the red half is more active. And so we see red after images. There's also a blue-yellow opponent channel. And so again, you see the same kind of after effects. And there's also a black-white channel here as well. And so this part of the processing is happening with the ganglion cells as they map on back to the LGN. And of course, even further back into V1 and V4. And we had that other flow chart previously that showed how some of the V4 neurons, let's go back there real quick, right here, give us color information that's part of that ventral stream that's taking us to the temporal lobe, right? So I just wanted to mention that color stuff real quick. But remember the movement, right? So movement is part of the dorsal stream, right? And this is going to take us into the posterior parietal cortex, where we're going to find what are called correlation detectors. And the correlation detectors are important for recognizing when an object is moving. So what we have here is we have two signals, maybe starting at the retina, for example. And what's happening is that if a neuron is moving some span across, a moving stimulus is moving across some span of the retina, we're going to call that span S, right? It covers some distance across the retina. And so it's going to activate receptors at location F1, and then it's going to move that distance, and then it's going to activate receptors at location F2. So F1 and F2 are both converging to some other neuron somewhere in the brain, right? So it says post-retina, so this could be anywhere in the brain, but we'll find these correlation detectors oftentimes as far back as the posterior parietal cortex, right? And that neuron now is called C2. So F1 and F2 both send a signal. So F1 is stimulated, right, by this stimulus that's moving across the retina. And then it sends a signal to neuron C2. F2 is also then stimulated by this moving stimulus, and it sends a signal to C2. But because F1 is activated first, because it's a moving stimulus, so it's going to activate these retinal receptors in a sequence, right? That means that F1 will send its signal to C2 before F2 sends its signal to C2. And if those onsets are too far apart, then we do not get our infamous temporal summation, right? And that means that C2 is essentially going to not respond to that signal that comes from F1 followed by F2. But notice here that there's a little box labeled T that separates F1 from C2. And this T is a delay. It's a delay box. So what's going to happen is that the signal from F1 is being delayed so that the time it arrives at C2 is maybe a half a second after the actual event occurred, right? Meanwhile, F2, half second later, is stimulated by this moving stimulus, and it sends its signal directly to C2. So now what's happening? Because F1 gets held back a little bit, F1 and F2 signals now arrive at C2 at the exact same time. right? Because we're going to use this delay box to prevent F1 from getting to C2 too soon. And that means that if this moving stimulus is moving at just the right speed that happens to match the delay that the box T imposes on the F1 signal, then those signals coming from F1 and F2 arrive at C2 at the same time, and we get technically a temporal summation, right? And in this case, C2 fires, but C2 only fires when the speed of that moving stimulus exactly matches the delay in that delay box T. What we might now imagine is that there could be many different such detectors called C2, C3, C4, right? Different correlation detectors. That's what the C means here. And each one is tuned to a different delay. And that means for shorter delays, those would detect faster moving stimuli. And for longer delays, those would detect slower moving stimuli. So each one of these correlation detectors is tuned to respond to a moving stimulus that has a differential velocity. So we have fast velocity detectors and medium velocity detectors and slower velocity detectors. And they're called correlation detectors because they are detecting the correlation in the timing of these signals, right? What we also find in the posterior parietal cortex are not just the correlation detectors, but also we find evidence of now these newly discovered mirror neurons. So mirror neurons, as it turns out, are specialized motion perception cells that are active both when we perform a specific movement with our bodies, but also when we observe another person perform that same movement. So if I'm waving, there are neurons in my parietal lobe that are active during that movement, but they're also active if I were to watch another person waving at me. So this obviously seems to suggest that there's some importance about understanding the nature of the movement of the body, both in yourself and in other people. An important part of social perception, perhaps, and social learning and observational learning is involved with these mirror neurons in the parietal lobe. So now this difference between the Watt channel and wear channel can help us understand some of these deficits that people may have with brain injury. So I mentioned agnosia already. Here are the different forms specifically. So if you damage those columns that are about recognizing objects, simple two-dimensional diagrams, that's called visual form agnosia. You can have color agnosia, by the way. It's also called achromatopsia, right? So now this is damaging your ability to perceive colors, but it's not the same as color blindness, right? Because it doesn't really have anything to do with the nature of the cones and how they work and whether you have 3 or 2 or what, but it's really deeper into the brain that causes this damage. And then again, prosopagnosia is the inability to recognize faces. But what's very also interesting about damage to the temporal lobe Watt pathway that may cause one of these types of agnosia is that your ability to interact physically with an object is not really impaired. So for example, you can draw the object from memory. You can reach for it and grasp it. So for example, if you look at the coffee cup again here in my little camera window here, if you have agnosia, you might not be able to identify what this is, but you can still reach out and grab it accurately. You can shape your grasp so that you can wrap your hand around it accurately. So you can still even see what it is and interact with it in a functionally appropriate way, even if you can't say what it is, right? And so this idea that you there's a separation between this kind of more explicit recognition task and the physical interaction task indicates the separate nature of these two visual pathways. And along those same lines, here we have research that looks at a patient who has an agnosia, and they use what's referred to here as the visual motor posting task. It's called a posting task because it's a bit like mailing a letter in a mailbox. You're putting it into the post, right? So in this case, in one version of the experiment, the person here is holding this rectangular shaped card. And when they are asked to simply just match the orientation, and so that means that they have to hold this card in a way that matches the orientation of the slot there, right? You don't have to put it in the slot. You just have to align it up so that they match. And what we see in the top half here is a circle with all these lines fanning out. That's really just a graph that shows the average of their performance, which is they're terrible at the task, right? If the line is presented, if the slot is presented vertically, they're not necessarily going to align that card vertically. They might align it any number of different orientations, right? And if it's presented horizontally, again, they're going to present any number of orientations. They just simply can't do it. But on the right are control subjects who are just neurologically intact, and their error rates are extremely low. They're very good at lining up the card to the slot. But in the posting version of the task, when you just tell this patient, put the card into the slot, we find this is patient DF. We're identifying her by the initials. What we find is that she can do it, right? She can put the card into the slot without any problem. Even though she cannot explicitly, consciously line it up when asked to just orient the card to the slot, when asked to just put it in the slot, she does so accurately, and she doesn't actually kind of like run it up there and twist it around until it goes in. She really does smoothly and accurately insert the card into the slot. So her motor performance is unimpaired, but when it comes to that conscious alignment of the stimulus to the target, she can't do it, right? And controls, of course, do well in both cases. Meanwhile, if you have damage to the dorsal stream, taking us from the occipital lobe up into the parietal cortex, this results in what's referred to as optic ataxia, right? So here, recognition occurs normally, but the ability to physically interact with an object is impaired. For example, reaching to pick up an object might become very, very difficult and very, very effortful as a person struggles to get their hand going in the right direction because they simply can't coordinate the visual information about location with their body to make their hands go where they want them to. All right, I think we're going to cover all of chapter 6 now at that point. So we are now good to go for that.